peptides, and proteins. Interesting chemistry. Okay, Chemistry 3111. This is our second to last chapter of the course, which deals with amino acids, peptides, and proteins. And as the name suggests, you can imagine that an amino acid has both an amino group in it. So an amino group looks like this, right? Everybody knows what a primary amine is. And then a carboxylic acid looks like this. So there are many compounds that contain both amines and acids in them. But what we're mainly going to deal with in this class will be the alpha amino acid. So I'll get into that. But before we do that, it says here that it might be necessary to review a couple of concepts before you start this chapter. First of all, the Cobb-Ingold Prelog rules, or how to designate the RS configuration to a chiral center. You can look at section 20.3 on carboxylic acids. Just a tiny bit of carboxylic acid chemistry shows up in here, but I'll go over that with you. Nucleophilic acyl substitution, that's also from chapter 20, and again, I'll cover that with you as well. And then the properties of amines, well, we just finished the amines chapter a few days ago, so that should be pretty fresh in your mind. Another thing I just want to point out about the whole RNS thing, because sometimes students find that intimidating, or they designate a stereocenter and they worry, oh, am I right? Is it the other one? Well, what we looked at in chapter 24 were all D sugars, right? We only looked at D sugars. However, when we're talking about amino acids, the only thing that we're going to look at are L amino acids. So L amino acids, because unlike sugars, the naturally occurring amino acids are the L amino acids. And so every amino acid is going to have an S configuration. Okay, they're all going to be S except one thing. And that one thing is cysteine. So I'll put here except cysteine. And that is actually one of the questions in the textbook. So now you know the answer to it. But we'll take a look at that as we move along. If you've ever wondered what a protein is, we talk about proteins. They became really popular a few years ago when the South Beach diet kind of took off, or the Atkins diet, and people said you need to eat a super high protein diet, no fats. Well, proteins are nothing more than compounds, like big molecules, that are made from amino acids linked together. So you can think of an amino acid as kind of like a puzzle piece. And then you link all these puzzle pieces together, all these different amide bonds that hold them together, and you can end up with a protein. And if you've heard the word enzyme before, which I'm sure you all have, an enzyme is nothing more than a protein, except it functions as a biological catalyst. That's all there is to it. So if you've heard of things like kinases and peptidases, those things that end in A's, those would be enzymes, and we'll talk about them very briefly at the end. But again, an amino acid contains an amino group, and you notice that if you zoom in here, you can see an amino group in the amino acid, and it also contains a carboxyl group. They all have a carboxyl group and an amino group. So amino acids, that's where the name comes from. But like I told you, there's many types of compounds that can have amines and acids present in them. However, the one that we're most interested in, or the ones that we're concerned about in this class, are the alpha amino acids. You've seen the Greek letters alpha and beta and gamma and delta show up many times in this class. So when we talk about alpha amino acids, if you think about, an aldehyde, you start at a carbonyl like this. You have 1, 2, 3, 4 carbons, right? This would be the alpha carbon. This would be the beta. This would be the gamma, so on and so forth. The gamma, there we go. So on and so forth. Well, if you have a carboxyl group in a carboxylic acid, and this is the carboxyl group, the carbon that's attached directly to it, that is the alpha carbon. So alpha, I'll just put it like this, alpha carbon. So when we talk about alpha amino acids, you have an amino group on the alpha carbon. So alpha amino acids. Now that alpha carbon, since it's got a carboxyl and an amine already attached to it, if it has an R group here that isn't a hydrogen, because there is a hydrogen here, okay, if it's got an R group on it, it's chiral, right? So all amino acids except for glycine are chiral. Well, if glycine isn't chiral, what's the only possible structure for glycine? Glycine must be this. It must be an alpha amino acid. So let me try to draw that a little neater, where you have a carboxyl, you have an amine, and you have two hydrogens here, like that. So put another way, you've just got this. That must be the structure of glycine because it has no chiral carbon in it. The alpha carbon, if it has any R group on it, whether it's a methyl or what have you, is going to be a chiral center. If you've ever heard the word peptide before, a peptide is nothing more than a polymer. It's a small polymer, I guess. It's a relatively short chain of amino acids linked together. So if you have anywhere, a couple of amino acids linked together up to 30 or 40, that is considered a peptide. And we'll look at some examples like vasopressin, oxytocin, bradykinin. These are all peptides, or glutathione even. Whereas a polypeptide describes a chain of fewer than 40 to 50 amino acids. So we can have a peptide, relatively short chain, and then we can have a polypeptide, so that can be a little bit bigger. And if you're wondering, what holds these peptides together? Well, this is something that's going to come up over and over and over and over in this chapter. What holds amino acids together in a peptide, protein, or enzyme are amide bonds. Everybody needs to know what an amide bond is. So if we have a carbonyl and then it's attached to a nitrogen, and you have R groups on here, that is an amide bond. I kind of ran out of space there, but that's an amide bond. And you see that we have an amide bond here, and we have an amide bond here. More specifically, where the arrow is pointing, that's the amide bond, the one between the carbonyl carbon and the nitrogen. We also call them peptide bonds. So instead of me calling it an amide all the time, when we say peptide bonds, we're specifically referring to an amide bond between two amino acids. I repeat, peptide means the same thing as amide, but when we're talking about amino acids, it kind of has its own lingo. And instead of saying, oh, this is an amide bond, if I say to somebody, hey, this is a peptide bond, the person knows I'm talking about an amide bond between two amino acids. In proteins, as I briefly mentioned before, are really just big biological molecules that have some kind of function in an organism. So let's talk about the structures of all of these amino acids. Before I started recording, I showed you the handout or the page of amino acids that I will give you for the quiz, or at least some of them. And there are 20 alpha amino acids that are naturally occurring. It says they differ in the identity of their side chains or their R groups. Now, if you think about human beings, what do we have in common and what makes us different? Well, most of our DNA is identical. There's very few things that make us different. And amino acids are kind of like, individuals in that way. What makes individuals unique? You could say it's maybe their spirit or something. Well, the spirit of an amino acid would be its R group. Now, if you're wondering why do we always talk about L amino acids, it's because the naturally occurring amino acids are the L amino acids. They're based off of L-glyceraldehyde, which the L actually stands for levorotatory, and L-glyceraldehyde. We looked at D-glyceraldehyde in the sugars chapter, but if you compare the chirality of these two naturally occurring amino acids, L-alanine and L-serine, just for example, you see that the amino group is on the left-hand side, like the hydroxyl is on the left-hand side of L-glyceraldehyde. And that's why we name them like that. Everything is based off of L-glyceraldehyde. But again, I repeat, the naturally occurring sugars were the D sugars, but the naturally occurring amino acids are not. They're the L amino acids. They're the opposite. All right, let's move on from there, and let's take a look at the different types of amino acids there are, because we're going to classify them according to their R group. We have a total of four subgroups that we can divide the amino acids into. The first ones are any amino acid that has some kind of nonpolar chain. Okay, so something that's just kind of like an alkyl group, doesn't have anything super, interesting. I don't know if that's the right word, but first of all, if you just have a hydrogen as your R group, that's glycine. We already saw that earlier today. Of course, there's another There's another hydrogen in there, right? They're not highlighting this one, but that would be glycine. Then we have alanine, where the R group is a methyl group. So this is just a methyl. In valine, it's an isopropyl. And again, you don't have to memorize all of these. I guess I repeated the valine here. Then we have a leucine. That's an isobutyl. So isobutyl. The isoleucine is actually a sec-butyl, but it's got a chiral center in it. But anyhow, we'll just put here sec-butyl. And I'm not going to quiz you a whole lot on the chirality of that. The next one is proline. And proline is a very unique amino acid because it's the only one where the amine is a secondary amine. It's the secondary amine. All the other amines, for example, like this or this or this, they're all primary amines. Proline's the only one that's different, which really gives it some interesting biological, properties. Then we have phenylalanine. This is a benzyl group here. This is called a benzyl. In tryptophan, this molecule here is called an indole. Like this portion here is called indole. And then you have a methylene, but I'll just write indole below this in case you want to look up indole for fun. And not only do we have the names of all these, but we have their 3-letter abbreviations and their 1-letter abbreviations. Again, you don't have to have those memorized, but you see that what all these R groups have in common is they're just alkyl. They're not polar. There's no oxygens. There's no opportunity for hydrogen bondings. The only thing that students will bring up sometimes is they'll say, well, what about indole? There's a nitrogen-hydrogen bond in there. Yes. However, in tryptophan, the reason this is considered nonpolar is because the R group is mostly nonpolar with just this tiny nitrogen-hydrogen bond in it. So it's considered to be nonpolar. So tryptophan, phenylalanine, proline, valine, leucine, isoleucine, glycine, and alanine. Next, we're going to look at amino acids that have polar side chains. These get a little more interesting. This one here is asparagine. Okay, its abbreviation ASN and N. So this has got a methylene here, and then you have a primary amide on the end. The next one is glutamine. You've got 2 methylenes, and then you have a primary amide on the end. You can see that these are polar, right? Because you have an amide on it, right? This has got a dipole on it going this way. This has got a dipole here. I don't need to go over all that at this point in the course with you. Serine, obviously this is polar. It's got a hydroxyl group right on it. Nobody argue with me on that hydroxyl is polar. Same thing with threonine. It also has a hydroxyl. Tyrosine has a hydroxyl on it. And cysteine has a thiol. So remember, the sulfur analog of an alcohol is called a thiol. So cysteine is also considered to be polar. Again, we have all their 3-letter abbreviations and their 1-letter abbreviations. Okay, so we've covered nonpolar and polar side chains. The last 2 subgroups, if you will, are the smallest groups, and these are the ones that have acidic and basic side chains. For acidic side chains, there's aspartic acid and glutamic acid. In aspartic acid, you have 1 methylene. In glutamic acid, you can remember it because it has 2 methylenes, and you might notice that there's a carboxylic acid on the end. Well, that stands to reason because it's called an acidic side chain. If you're very astute, and I know you all are, you probably noticed that aspartic acid is nothing more than the carboxylic acid analog of asparagine, meaning if you take this and you put a hydroxyl here, you would have aspartic acid. So asparagine and aspartic acid are very closely related. Same thing with glutamine. If you were to put a hydroxyl here, you would have glutamic acid. So glutamine and glutamic acid are very closely related. The last 3 have basic side chains. We have arginine, which is this really interesting functionality on the end. Histidine, which incorporates an imidazole ring. So this is called an imidazole. You might remember that from the chapter on aromatic chemistry. And then a lysine. This is the most obviously basic one because it has an amine just dangling off at the end. It also has 1, 2, 3, 4 methylenes before you get to that amino group. So there you go. Those are all the naturally occurring amino acids, all 20 of them. If you're the kind of person who likes to peruse the internet, you will notice that this is not an exhaustive list. Okay, if you Google the common amino acids, these are the ones that are going to come up in any textbook, any YouTube video. However, these are not the only amino acids that are out there. There are all kinds of naturally occurring amino acids. These are the 20 most common. These are the ones that come up all the time. When you take biochemistry, you're going to run into a whole bunch of other amino acids. But again, these are the ones that we're going to stick to in this class. Now, something you might have heard of before, especially if you studied biology, is essential amino acids and non-essential amino acids. Well, it turns out that we can only make half of the amino acids ourselves. Half of the amino acids can be built by our bodies, whereas the other 10, the only way to get them is to eat them, is to consume them, to get them from your diet. So you might have heard of essential protein or complete protein sources. Complete protein are foods that contain all 20 naturally occurring amino acids. Examples of complete protein would be things like meat, fish, milk, eggs. Those have every bit of amino acids that you would ever want. However, no single plant source contains all of the essential amino acids except soy. Okay, so I'll put here except soy does have all the essential proteins. So that's why in every biochem and every organic chemistry textbook always has this blurb here where it says if you're vegetarian, you have to mix a combination of grains, beans, nuts, etc., rice, to get all of the amino acids. For example, if you've ever heard of the dish succotash, it's a mixture of beans and rice is deficient in 2 amino acids, and beans is deficient in 2 amino acids. But when you put them together into a delicious dish, you end up with a source, a homemade source of complete protein. So there you go. Something kind of interesting there. Like I told you, there's all kinds of amino acids that aren't alpha amino acids. For example, GABA, which is a neurotransmitter found in the brain, gamma aminobutyric acid. It's an amino acid, but it's not an alpha amino acid. Or thyroxine, which is found in the thyroid gland, is also an amino acid, but it's not an alpha amino acid. So there's all kinds of different examples. If you've ever heard of para-amino benzoic acid, PABA, that would be another example of an amino acid that's not an alpha amino acid. I can write the acronym here if you want to look it up. I won't be quizzing you on this, but if you want PABA, that's also an amino acid. Well, you can imagine that if the whole chapter is about compounds that have both an amino group and an acid in them, we know that amines are basic. We know that acids are acidic. Acid-base chemistry has to be a part of the chapter, and you would be right in assuming that. So let's take a look at the pKa values of the amino group and the acid group in an amino acid. It says under acidic conditions, so a super low pH, you're going to protonate everything in the molecule. Okay, if you have an amino acid, where's the most protons you can put? I mean, the carboxyl group can only handle 1, but we know that we can make the ammonium ion of the amino group. So this would be called completely protonated. Give me a thumbs up if you follow me on that. Just everything here is completely protonated. You're at low pH. It's very acidic. So very acidic. Okay, cool. I just want to make sure you follow with me on that because we're going to build up to something a little more complex. So the proton that's highlighted in red, it has a pKa, and the proton highlighted in blue, it has a pKa. They both have their own unique pKa value. Somewhere in the middle, you end up with a situation where you have an anion on here and a cation on here, and we call that, and you remember this, a zwitterion, right? Zwitterion is a German thing. Zwitter is like, I don't know, like the two of them are existing in the same molecule. So you end up with something that's neutral, the zwitterion. But if you put it in a very high pH, so something really basic, that's going to rip off all the protons, and then you end up with something that's negatively charged. Whereas if you put it in a low pH, something super acidic, it's going to protonate everything. And like here, you're going to end up with something that's positively charged. All right. So let's take a look at those pKa values, the pKa for the carboxyl group and the pKa for the amino group. First thing is if you look at the pKa values for the carboxyl group, you can see very quickly they're kind of all in the same range. they're all around 2. So they have a pK around 2, 2-ish. It's a little above, a little below, but 2 is close enough. Whereas the amino group, it's around 9, 10. So we'll put here 9 to 10-ish. But those are both very close. So the carboxylic acid is more acidic than the ammonium ion, but that shouldn't really be a surprise. Okay, if you go back to the Areo principles, it makes sense that it would be more acidic, and the pKa values don't lie. So what I would think you would want to know as a student in the class is just have a ballpark idea. Generally speaking, the carboxylic acid is around 2. And generally speaking, the amino group in my amino acid has a pKa when it's protonated of about 9 to 10. All of these other pKa's here, and this isn't an exhaustive list of all the amino acids, but those are the pKa's of the side chain. For example, let's say you have aspartic or glutamic acid. Well, their pKa's of their side chains are pretty low as well because they're carboxylic acids. Whereas if you take something like arginine, which is basic, its pKa of its side chain is really high, right? Because it's a basic side chain. Let's move on from there. Acidic and basic side chains have their own pKa. You see for tyrosine and lysine, those are thrown in here. Anyhow, let's not worry about the side chains right now. Let's just focus on the carboxyl group and the protonated form of the amine and see if we can unpack a little more of the information here. And let's talk about physiological pH. Could anybody tell me what physiological pH is? Ballpark? Maybe better than a ballpark, but what would you say physiological pH is for my students who have studied biology? I'm sure you know. Yeah, it's around 7.3. Yeah, 7.4, somewhere like that. So you see that physiological pH lies in between all those pKa values, doesn't it? It lies in between the carboxyl and the amino group. So what does that mean? The pH of the human body is more basic than these carboxyl groups, but it's more acidic than these amino groups. So what does that mean? Let's take a look. What it means is that at physiological pH, since physiological pH is around 7, where do I have it here? Physiological pH, we'll put here around 7 point, we'll put 7.3 to 7.4, something like that. So what that means is if the pKa of this is equal to 2-ish, okay, let's say, well, this is more basic. So that means it's going to deprotonate it, and at physiological pH, the carboxylate form of an amino acid is going to predominate in the human body. Conversely, if we look at the amino groups, okay, at physiological pH, since you're at 7.3, 7.4, and all of these are around 9, well, you're in a medium that's more acidic than these, so these are all going to be protonated, the alpha aminos, the alpha amino groups. And that is why at physiological pH, so in the body, the structure of an amino acid is going to be as zwitterion. You have the carboxylate and you have the protonated form of the amine. Give me a thumbs up if that just makes some sense to you. If you're like, I'm like 70% of the way with you or even more. If it's normal for people to have to go back and look at things. I would think it'd be pretty crazy if you didn't have to. Okay, it's totally normal to go back and review. All right. So we've got our amino acids in the human body existing as zwitterions. Well, let's take a look at this physiological pH thing a little bit more. It says at physiological pH, amino acids are amphoteric. That means that they can function as an acid or a base. If you have the zwitterion form of an amino acid, well, if you put it in a strong base, it can rip the proton off the protonated amine. But if you put it in a strong acid like this, then you're going to protonate the carboxyl group. So again, they can behave as an acid, as it is here, or they can behave as a base, as it is here. Well, it's behaving as a base. Amphoteric, functioning as an acid or a base. Now, the next thing I want to cover is what's called the isoelectric coin. And I'm sure that somebody who's in this lecture has performed an electrophoresis experiment in a biology class. I know I did in my first year biology class 27 years ago. So I do remember doing that. you know, setting up the electrodes and everything. I don't remember understanding it completely at the time. I figured it out after I studied organic chemistry. But we take advantage of those pKa values of the amino acids to enable us to separate them. So let's take a look here. It says the isoelectric point, Pi, is the pH, specific pH, at which the Zwitter ion concentration is the highest. How do you calculate that isoelectric point? All you do is if you have an amino acid that doesn't have one of those extra side chains, you just take the average of the two pKa's. That's all you do. So let's say we have something that has a pK of 2.34 for the carboxyl group, 9.69 for the amino group. All you do is take the average and you say your isoelectric point, Pi, is 6.02. So if you have this amino acid in a solution that is exactly at a pH of 6.02, you're going to have the maximal concentration of the Zwitter ion, where this is protonated as it is here, and this is going to be deprotonated. Okay, maximum concentration of the Zwitter ion. That's the isoelectric point. If you're wondering, well, what the heck do I do if I get one of those amino acids that has one of those funky acidic side chains or a basic side chain? You know, what do you do with those? Well, in those cases, you take the average of the two pKa's that are closest. So let's say you have lysine. Well, lysine is basic. This is the basic pKa. This is the other basic pKa. You take the average of those. If you're in a solution that is having a pH of 9.74, you have the maximum amount of the Zwitter ion, where everything is perfectly balanced as a Zwitter ion. If you have an acidic side chain, or two of them, like in glutamic acid or aspartic acid, you just take the average of those two acidic side chains, and there's the isoelectric point for that. So the way that electrophoresis works is it takes advantage of the isoelectric point of amino acids. Think about this. If the isoelectric point of the amino acid is greater than the pH, so that means the pH is lower, then you're in an acidic, or it's basically going to be protonated. So that means it's cationic. If you put it in some kind of vessel where you have a cathode and an anode, if it's cationic, it's going to drift towards the cathode, right? Because it's positively charged, it's going to drift towards the negative end. Conversely, if you have an amino acid that has an isoelectric point that's lower than the pH, that means the pH is higher than its isoelectric point. That means it's going to exist as an anion. Well, in that case, it's going to drift towards the anode, the positively charged end. So again, we're just exploiting the isoelectric point of an amino acid. Let me just draw glutamic acid very quickly for you. So if we take a look at glutamic acid, so glutamic acid is this amino acid here. Anyhow, I won't draw so look, let's say we're at pH 6. That means that this proton is going to be ripped off. So we're not going to have this proton. Then this proton is also going to be ripped off because we're at a pH, those two are going to be, what am I doing wrong here? No, those two are going to be ripped off because we're at a pH that's more basic than the pKa's of the two carboxyl groups. Then this part is going to be protonated because we're at a pH that's lower. That means it's more acidic than the pK of the amino group, but these two charges cancel, positive and negative. So overall, this amino acid has a charge of -1. So if it's negative, it's going to drift towards the anode. It's going to drift towards the positive end. Does that make sense to everybody? Give me a thumbs up if you're with me on that. Just the whole concept of the isolate. Okay, cool. Yeah, it's, and another thing, something that I never figured out until I took organic chemistry is I said, okay, well, what if you have two amino acids that have a very similar isoelectric point? You know, are they really going to separate that much? Well, it's not only like electrophoresis, it's not just based on charge, it's also based on molar mass. So let's say you have two amino acids that have very similar isoelectric points. The heavier amino acid is going to move slower. It just because it's heavier, it's not going to move as fast. It's going to take more kinetic energy, I guess, to make it move. Oh, I have it written down here. I didn't even realize. Okay. The larger amino acid moves more slowly. All right, good enough. So that's a little bit about electrophoresis. I don't know. I would think it's kind of interesting to talk about that. But electrophoresis, really the purpose of electrophoresis is just to separate small amounts of amino acids and find out how many amino acids you have in a mixture. When it says analytical scale technique, analytical chemistry means chemistry dealing with very small quantities. Okay, tiny little quantities. It's not my, not my favorite thing in the world to deal with little tiny quantities. The quantities that we use in the organic lab at UCCS are small enough. So we're talking like very tiny quantities. But amino acids, like they're coming out in your sweat right now as we speak. Like your fingertips are covered in sweat with amino acids on them. And those amino acids, I mean, if you sweat and you touch a piece of paper, you don't see anything, right? When you're spreading your amino acids around a room or whatever. So they're colorless, but they can be detected with what's called ninhydrin. If you've ever watched any of those crime shows where they're looking for fingerprints, you might have heard of ninhydrin stain. They can, you get a purple colored product. Has anybody ever seen that at a crime show? They spray this spray, not luminol, that's something else, but they'll spray it on fingerprints and it turns purple. Anyhow, our book seems a little obsessed with ninhydrin, drawing the product of ninhydrin. But the bottom line is you just end up combining the amino acid with this molecule called ninhydrin, and you end up forming a purple stain, and then it liberates, part of the amino acid as an aldehyde, and you get some CO2 plus water. Let's try a question here. Let's take a break from learning stuff and try it a different way by trying a problem. It says, although most naturally occurring proteins are made up of only L amino acids, proteins that are isolated from bacteria sometimes have D amino acids in them. Draw Fischer projections for D and alanine and D-valine. In each case, assign the configuration of the chiral center as being R or S. So let's start with L-alanine. Why don't we start with that? So if you remember, if we had D-glycine, and this is something that I think it's a good idea to remember back from your carbohydrates days, this is D-glycine. So if the hydroxyl on the right-hand side, it's D. So if we want to make, did I say D-glycine when I meant D-glyceraldehyde? Sorry. It's not D-glycine. Glycine isn't chiral. So this is glyceraldehyde. Sorry about that. All right, so we have D-glyceraldehyde. So now we know which side the hydroxyl points on for glyceraldehyde. So the way that we draw an amino acid is similar. We have the most oxidized carbon at the top. What am I doing? There we go. Where's my red pen? Come on. There we go. Son of a gun. Okay, there we go. So we have our most oxidized carbon at the top, CO2H. We have this line going down here. We have a line going like this. And since we have D-glyceraldehyde, we're going to put our amino group over here, NH2. Then we're going to put our hydrogen down here, and our R group is going to go down here. So our R group in alanine is a methyl group. So this is alanine. And we know it's D because we have the nitrogen on the right-hand side. So that designates that it is a D amino acid. So this is D-alanine. Now, at physiological pH, it wouldn't exist like this. It would exist as this Zwitter ion, so we could remove the proton from here and make the carboxylate and make the protonated form here. So D-alanine. Now, if we want to make D-valine, what's the R group in a valine? So the R group in alanine was a methyl group. The R group in a valine was an isopropyl group. So all we have to do is something very similar Put the NH3 group over here, put the carboxylate at the top, and then we have an isopropyl down here, CH with 2 methyl groups coming off of it like that. And that would be D-valine. Again, neither of these are naturally occurring amino acids. However, they are amino acids. Now it's asking us, are these R or are they R or are they S? Well, let's take a peek. Let's start with alanine. We look at the 4 atoms. We have a carbon, a hydrogen, and a nitrogen. The nitrogen has the highest atomic number, therefore it must be number 1. The hydrogen is going to be last place. The carboxyl is a higher priority than the methyl. So we have 1, 2, 3. We're going counterclockwise, which would be S. But remember that this proton, since it's horizontal and a Fischer projection is coming out towards you, so we reverse it, and this would be R. So we have the stereochemical designation there. We do the same thing here. We go from 1 to 2 to 3, which should be S. We reverse it. It's R. So both of these have R stereochemistry. Now, these are both Fischer projections, and that's what the question was asking for. It said, can you draw a Fischer projection? There's nothing wrong with a Fischer projection. However, oftentimes we're not going to draw the amino acids this way. Oftentimes, I like to draw them like this, where I have the carboxylate like this, and then I have the amino group coming down like this. So for alanine, here's my methyl group over here. And the reason I have the nitrogen out in front is because that means the proton is in the back. I go from 1 to 2 to 3, like that. What did I do wrong here? Oh, I drew the natural form, so I need to draw the unnatural form. My mistake. There we go. So like this. So this would be D-alanine. This is the unnatural form. And you could do the same thing with D-valine. All right, let's move on and let's try another question. This is question 25.2. It says, draw the form of the amino acid that's expected to predominate at the stated pH. So we're starting out with alanine at a pH of 10. Here's what we're going to do. I'm going to write out the structure of alanine in its fully protonated form. So alanine, as I just tried to draw it, is like this. And we'll put the protonated form of the amine here. I've gone and looked up the pKa values in the table. So the pKa of the carboxyl group is 2.34. And the pKa of the amine, the protonated form, is 9.69. So if we're at a pH of 10, this is more basic than both pKa values. Well, if we're in a more basic environment than either of these pKa values, that means it's going to be completely deprotonated. So at a pH of 10, we're going to have the carboxylate, not the carboxyl, and we're just going to have the unprotonated form of the amine. Give me a thumbs up if you follow me on that rationale. Are there any questions about that? It's just a matter of assessing the pKa's, which you get from the table, and then you have to understand that a high pH is more basic, a low pH is more acidic. Okay, cool. Let's try proline. Same rationale. So I'm going to draw the whole proline molecule. Just bear with me here. Like this. So I'll do something like that. And I'll draw the protonated form of it. Remember that proline is a secondary amine. It's the only one. The pKa of the protonated amine is 10.60. And the protonated form of the carboxylic acid is 1.99. If we put this in an environment that's a pH equal to 10, it's going to be more basic than the carboxylate, so it's going to deprotonate it, but it's more acidic than this one, so this one is going to stay protonated. Ergo, it's going to look like this. So this is going to stay in its protonated form, and the carboxylate is going to be deprotonated. The next one is glutamic acid. So this one has 3 pKa since it's got an acidic side chain. Let's write the whole thing out. We've got time. So here's the first carboxyl group. Here's the nitrogen of my, there we go. So glutamic acid has 1, 2 methylenes. Then it has another carboxyl group here. Okay, the pKa of the most acidic one, its pKa is 2.19. The pKa of the protonated form of the amine is 9.67. Again, these values come right from the table. You don't have to memorize any of these. The side chain is 4.25. If I put it in an environment with a pH that's equal to 3, that's going to be more basic than this one. However, it's more acidic than both of these. So those two that I have the circle around are going to remain the way they are. It's only the one here that's going to be deprotonated. So let's draw that form of the molecule. It's going to look like this. So this one stays protonated. This nitrogen stays protonated. And it's only this carboxylate that's going to lose a proton like that. All right. What do you think? Not too bad? Well, if you want to bring a calculator into the quiz to calculate isoelectric points, I don't have a problem with that whatsoever. It says use that same data table to calculate the isoelectric point of the following amino acids. I don't think we need to write out the structure of these. We just really need to look up their pKa values. So if you look up aspartic acid, you see that the carboxyl group attached to the alpha carbon has a pKa that's 1.88. The alpha amino, when it's protonated, has a pKa of 9.60. In the side chain of aspartic acid, it does have a side chain. It's got a carboxylic acid. Its pKa is 3.65. Now remember, when you have a side chain, you take the 2 pKa's that are the closest to calculate the isoelectric point. And thus, our isoelectric point, Pi, is going to be equal to 1.88 plus 3.65. We divide that by 2, and we end up with 2.77 as being the isoelectric point. Let's just do one more. I'll let you do lysine on your own, but let's just try leucine. Leucine does not have a side chain. It just has the alpha carboxyl, which has a pKa of 2.36. It has the alpha amine. Its protonated form has a pKa of 9.60. So to calculate the isoelectric point, you just say 2.36 plus 9.60. Divide that by 2 gives you 5.98. The last one, lysine, if you look it up in the solutions value, the Pi, or the isoelectric point, is 9.74, which is pretty high. And that makes sense because lysine has a basic side chain. And so you would take the average of the basic side chain and the protonated amine. All right, I'll let you try that one on your own time.